Hi, everyone. It's good to see you again. Before we start today's talk, I'd just like to acknowledge my co-authors. This was truly a collaborative project with different folks taking different leads on different portions of it, and it wouldn't have been possible without these huge efforts from everyone involved. So thank you. So a brief background on Cisco and the lake. In the first part of the 20th century, Cisco were an important component of commercial harvest in Lake Michigan. But due to a likely combination of overharvest, invasive species, and environmental degradation, stocks collapsed by 1960 and were largely lost from the lake. While Cisco were not extirpated from Lake Michigan, they were extremely rare in the basin in the late 20th century. However, Cisco started to expand again in northeastern Lake Michigan around 2011 as ecological conditions changed. This is easily observed in multiple data sets, including commercial tribal harvest, shown here, as well as recreational harvest and fisheries independent data in the Great Lakes. As Cisco abundance has increased, we have also seen increases in angler catch and the establishment of recreational fisheries. This has largely been centered in northeastern Lake Michigan near Grand Traverse Bay. Of note is the large portion of the Cisco population that is reaching much larger sizes than historically reported, with anglers catching fish commonly around 19 inches or 500 millimeters in length. This suggests these fish are reaching sizes that are larger faster than historically in the lake or currently in other Great Lakes systems. Likely contributing to this higher growth is that contemporary Cisco and Lake Michigan are largely feeding on fish, a shift from a more planktivorous diet that was observed in historic populations of Cisco and contemporary populations in other Great Lakes. You can learn more about this change from a recorded webinar by my co-authors, Jory Jonas and Ben Tershak, which is available on the Great Lakes Fishery Commission's YouTube site. So as the fishery has grown in the main basin, we have also observed increased catches in some connected waters in Lake Michigan, specifically unique coastal habitats known as drowned river mouths. Drowned river mouths are transitional zones between the Great Lakes and tributaries and were formed following glacial retreat, where melting river water pooled behind glacial till, eventually carving out channels to the main basin. They are highly productive environments that are particularly common along the eastern shore of Lake Michigan, the region in which we have seen Cisco expanding in recent years. There are over 24 DRM or drowned river mouth lakes along the eastern shoreline, many of which, as denoted in this map, have lake-like habitats that are navigable by boats and can provide important seasonal habitats for Great Lake fishes, such as yellow perch, sturgeon, and alewife. Of special interest to today's talk on Cisco migrations is the northernmost DRM shown, Lake Charlevoix. Lake Charlevoix is the largest DRM lake in Michigan with an area of 69 square kilometers and a maximum depth of 37 meters. It's an oligotrophic system and it's connected to the main lake after passing through Round Lake and the last portion of the Pine River. It's in this connected channel that we have seen an emergence of a popular seasonal fishery for. During a short period in May and June, thousands of Cisco are harvested in the channel or nearby, providing anglers with a great recreational opportunity. While some Cisco are captured in other connecting channels, catches are orders of magnitude lower than what was observed on the Charlevoix Pier. It's often assumed these fishes are in the channel and the surrounding area for foraging. In addition to the spring fishery, a strong winter fishery has emerged on Lake Charlevoix. Ice fishing for Cisco has become popular on Lake Charlevoix over the last decade, with dozens of shanties on the ice on most days. Anglers are rewarded with large Cisco to take home, and guided ice fishing trips are also becoming more common. Despite its popularity, we still don't know a lot about the fishery, specifically if these fish are truly migrants to Lake Michigan, residents of the lake, or some mix of both. This has relevance to management of the fishery by Michigan DNR, as bag limits have been adjusted in recent years under the assumption that these fish are part of the broader Lake Michigan population. Additional information on movement patterns and determining whether this is any evidence of a resident population or a mixed population of the lake would have clear benefits for management. And Lake Charlevoix isn't the only DRM lake where Cisco are being harvested through the ice. Reports from 6 other DRMs are present in the Michigan Master Angler Database since 2017, as far south as Muskegon Lake. Understanding winter ecology and other DRM lakes can be useful in a broader understanding of seasonal habitats for the species, but fish concentrated in these habitats may also offer us an opportunity to explore questions that are related to the recovery of Cisco in Lake Michigan overall, and specifically whether these populations expanded from a single or multiple stocks. There are two primary hypotheses on how Cisco have been expanding in Lake Michigan. As postulated in a recent review by Claremont et al., the first is what we could be referred to as a single stock hypothesis, where Cisco have expanded from a small stock in Grand Traverse Bay and are repopulating Lake Michigan, mostly along the eastern coastline. An alternative is that there might be multiple small remnant stocks in Lake Michigan that have emerged as conditions improved in the lake, potentially from southern Lake Michigan, northern Lake Huron, or even the connecting waterways. While existing catch data suggests the single stock hypothesis is more likely, looking at genetic structure from these samples in DRM lakes could provide us an opportunity to explore this hypothesis while these fish are relatively easy to catch in different areas of Lake Michigan. With these ideas in mind, my co-authors and I received a pilot study grant from the Great Lakes Fishery Commission to answer several questions related to Cisco use of DRMs in the winter months. Today we'll touch on three of these questions. The first being, are seasonal movements at Lake Charlevoix Cisco indicative of migrations from Lake Michigan, resident populations, or both? Secondly, is there a genetic or morphometric differentiation of Cisco in Lake Michigan that suggests the recovery of multiple stocks? And finally, what is the feeding ecology of Cisco in drowned rivermouth lakes during winter months? We were able to take advantage of two existing acoustic arrays in northeastern Lake Michigan to address our first question on seasonal movements of Cisco in Lake Charlevoix. These arrays are supported by different studies funded by the Great Lakes Fishery Trust, Great Lakes Fishery Commission, and the Great Lakes Acoustic Telemetry Observation System. In the winter of 2022, we installed 3 additional receivers in Lake Charlevoix proper to track movement within the lake, and there are 2 existing receivers in the Round Lake system near the Pine River Channel entering the main basin. Around the same time, we spent several days on the ice implanting Cisco with acoustic telemetry tags in the middle of Lake Charlevoix. We did this using our traveling tagging station, which was set up using a heated ice shanty and had continuous flow over fish during surgery. We also aerated a recovery tank on the ice, and fish were collected via ice angling, and we were able to implement 20 Cisco with acoustic telemetry tags and collected an additional 25 fish for gut analysis, stable isotope analysis, and morphometrics. A total of 71 fin clips for genetic analysis were also collected, and we were able to increase the sample size by using fish donated from anglers that were out on the ice fishing with us. And I'd just like to note that a lot of the success associated with this event was really due to one of my co-authors, Ben Tershak, who did all the legwork to make it happen. Because we were surrounded by interested anglers, it was a pretty rewarding experience overall, where we got to show off some of the work we were doing and why. Before we jump into the results, I'll note that 14 of these fish survived long enough to be tracked during this period, with 2 likely dying as a result of tagging and 4 likely collected by anglers in the early spring. That would suggest a tagging survival rate of 70 to 90%, which is really quite high, albeit a very small sample size, so we were happy with those results by using ice-angled fish. So here we have the acoustic array in the main basin on the left and Lake Charlevoix on the right. We were able to track these fish over a year and into the spring of 2023, and further for some fish into 2024. As I play this animation, you'll begin to see changes in the colors of the receivers on Lake Charlevoix, which is indicative of fish movement. And as we get further into the early spring in May and June, we see these fish move out into the main basin, moving sporadically throughout the receiver array and potentially beyond it, and also into Grand Traverse Bay. And as we approach the spawning season, in early October into November and December, we see these fish aggregating near Elk Rapids, potentially to spawn, before exiting and moving back into Lake Charlevoix by December, at which point they stayed in the lake through this first year of tracking into late May. So overall, it appears that Cisco are using Lake Charlevoix as overwintering habitat. When we look across the entire set of 14 fish over a 2-year period, we can see some interesting points. The first is there appears to be uniformity in movement patterns among these fishes. Here we have a location of occupancy divided into Lake Charlevoix in yellow, the main basin in turquoise, and Grand Traverse in purple. And the importance of Lake Charlevoix is clear as these fish are spending 5 to 7 months of their lives in the water body. We also see strong site fidelity, not only for spawning in November and December in Grand Traverse Bay, but also for Lake Charlevoix, as all of our fish return to the lake in 2023. Moving into 2024, 10 fish retain this pattern of movement with strong site fidelity for another season, while 4 more were lost due to angling or end of the tag life. While we were somewhat surprised by these results, They actually align with a recent study out of Lake Ontario, where Cisco had strong site fidelity for not only spawning, but also non-spawning habitats throughout the year. So again, the primary message here is that Cisco appear to be part of a larger spawning aggregation in Grand Traverse Bay and are using Lake Charlevoix as overwintering habitat, and we don't see any evidence of resident population. In addition to tagging fish in Lake Charlevoix, in the winter of 2022, we were collecting fish from 2 other drowned river mouths where anglers had reported catch of the species. And this was to address our second question of whether there was any evidence of unique stocks in DRM lakes or along the eastern coastline of Lake Michigan. The 2 lakes we focused on were Portage Lake, which is about 1/8 of the size of Lake Charlevoix, and further down the coast, Muskegon Lake, which is about 1/4 of the size. These fish were collected by our partners at NOAA either directly or from angler donations. For our examination of genetic structuring, we included 2 other sources of fish from Lake Michigan. The first was fish collected from the Ludington Barrier Net Monitoring Program in the main basin. We included fish from the earliest part of the sample season, April and May, as this appeared to represent a period of time where the fish from Lake Charlevoix hadn't yet moved into the main basin based on our telemetry data. We also included 41 samples from the Grand Traverse Bay spawning aggregation that was collected in 2022, as we are interested to see if there was any evidence of them being the same genetic stock as the apparent migratory fish collected in Lake Charlevoix. We used a genetic technique called RAD-seq to examine genetic structuring, which subsamples across the genome. We then used a multivariate technique, principal components analysis, to try and visualize whether Cisco grouped by collection sites. If there were any genetic distinctness between our collection sites, we would anticipate samples from the same site to group together in multidimensional space. We also used principal components analysis to examine how samples from a nearby stock of Cisco near Boot Island in northern Lake Huron compared genetically to Lake Michigan fish. So essentially, we treated them as an outgroup to see where they landed in multidimensional space and genetic distinctness. In addition to examining genetic structuring, we also conducted morphological analysis using the measurements described in Eschenroder et al. 2016 in his monograph on Cisco's in the Great Lakes and Lake Nipigon. Specifically, we measured 9 linear morphometric characteristics, and we counted the number of gill rakers on the first arch of each fish. We then used a size-corrected approach to remove allometric effects. For this analysis, we focused on Cisco in the 3 DRMs, as well as those collected outside the Ludington pump stage plant that were collected in the early spring, again, prior to that date where we saw Cisco moving out of Lake Charlevoix. We were only able to conduct this for fish where we had whole body specimens in each lake, with sample sizes shown here on the right. We then again used principal components analysis to examine potential differences in morphology that might be indicative of independent stocks across the 4 study sites. So what did we see? In a nutshell, we didn't see any evidence of genetic structuring across the fish collected in DRM lakes, Grand Traverse Bay, and the main basin in the early spring. You can see our 2 PCA axes here that we just basically have a multicolored shotgun blast as opposed to grouping by region. Now, when we add in that presumed unique stock from Boot Island, we can see that the Boot Island fish, shown here in blue, cluster together along the first PCA axis, distinct from everything else we collected on Lake Michigan in 2022. This analysis also suggests that there's quite a bit more genetic variation in the Boot Island stock versus these fish collected on Lake Michigan, which had relatively low heterozygosity overall. So all in all, the genetic data set appears to support the single stock hypothesis of an expanding Cisco population in Lake Michigan. When we examined the PCA of the morphology dataset, we saw little evidence of distinct groupings among the 3 DRMs and the fish collected from outside of Ludington. There was some distinction associated with greater body depth in Lake Charlevoix and some smaller fin measurements in Portage and Muskegon Lake, but we do not think these differences are particularly compelling, especially given the small sample sizes in those 2 systems. Moving on to the final question we wanted to address today, we were really interested in understanding what exactly Cisco are eating in drowned river mouth lakes during the winter, and if it varied from what's been observed in existing data sets in other regions and seasons of Lake Michigan. Cisco diets vary seasonally and are predominantly composed of 3 invasive species. Based on available diet and stable isotope data, they seem to target goby in high abundance in the spring, alewife during the period of stratification in summer months, and spiny water fleas in the fall, with other diet items like chironomids and other fish making up a smaller portion of their diets at specific times of the year. What's not well understood is winter and early spring diets on the lake. And although there is anecdotal evidence from anglers suggesting they're targeting alewife in the DRM lakes, we really didn't have any hard data for that. So looking at the stomachs of the fish that we collected in the winter of 22, we see that Cisco seem to be primarily feeding on fish and most notably alewife. Here we have mean wet weight of diet items expressed as a percent from Cisco stomachs. We observed only fish in all of the stomachs that we looked at during the winter months. And Lake Charlevoix and Portage Lake were nearly entirely alewife. In Muskegon Lake, where we owned only 6 stomachs with fish in them, 7 were empty, shown there in parentheses, alewife were just under 50% of the mean weight, while other species like rainbow smelt, shiner species, and brook silver size were also in the diet. And there was a large number of unidentified fish as well. In addition to looking at what they were eating, we also looked at how full the stomachs were across the 3 DRMs using index of fullness, which is really just a ratio of stomach weight to total weight of the fish. And the main message here is that Cisco are eating significantly more in Lake Charlevoix compared to the other 2 systems, with stomach weight approaching 1% of the total fish weight of some fish. So wrapping up with just a few take-home messages. First, Lake Charlevoix provides overwintering habitat for Lake Michigan Cisco, and they actually have occupancy in some cases for nearly half their lives. They're likely part of the Grand Traverse spawning aggregation, and there really is no evidence of resident spawning populations in the lake. We also saw no evidence of unique remnant stocks along the eastern shore of Lake Michigan, and this does support the single stock hypothesis presented in Claremont et al. 2019. And finally, overwintering Cisco are consuming fish, and most notably alewife in large numbers while they're in these DMR lakes. In terms of next steps and future directions for this work, we're hoping to increase monitoring of Cisco overwintering in DRM lakes in the upcoming years through some additional grants. And we have a few primary questions. Are occupancy patterns in other DMR lakes similar to what we see in Lake Charlevoix? And additionally, are Cisco overwintering in southern DRM lakes and spawning nearby? And this is really of interest, whether we can identify whether these are all fish coming from Grand Traverse Bay or whether there could be localized spawning that has been establishing in southern Lake Michigan. And additionally, we're hoping to continue on with our stable isotope analyses from the fish we collected in this study, really disentangling a shift in diet versus a shift in habitat use. For instance, carbon signatures that we took from Cisco liver and tissue are more depleted in Lake Charlevoix relative to Muskegon Lake, and generally are on the extreme end of stable isotope values that we see in the main basin overall. And this really supports observations from the diets in Lake Charlevoix that Cisco are feeding heavily on alewives while they're in the DRM, and that they might be a resident population of alewife that are the primary food resource. So we are currently also looking at stomach contents collected from these diets to test this hypothesis by comparing their isotope signatures to those from the main basin that were collected in the northwest portion of the lake. We're also using stable isotope analysis to see what we can understand about species diet and movement from another tissue, the islands. This ongoing work is being led by our co-author, Willie Fetzer at the University of Wyoming, who has used the islands to track changes in stable isotope signatures over the entire life of different fish species. The islands can be peeled back like an onion, with each layer indicative of a different period in the fish's life. In the figure on the right, the core of the islands is represented by 0, with each subsequent layer of the eye representing a distinct feeding period later in life, until the most recent period just prior to capture. So for the first 2 fish in the figure on the right from Lake Charlevoix and Muskegon Lake that are of similar size and age, we can see some pretty definitive periods. First, a period where carbon signatures are not changing dramatically, perhaps through the first 1 or 2 years of life, consistent with isotopic spaces that we see on Lake Michigan. Then a period of time when the signature rapidly declines, then back to a heavier signature before potentially declining again. These changes could in fact be representative of transitions in diet that are associated with movement into DRM lakes. The first dip may be in the 2021 winter period prior to sampling, and the second dip, the beginning of the new drop in isotopic weight associated with the fish moving back into the DRM and foraging isotopically on lighter prey. Because isotopic signature changes with somatic growth, it seems as though DRM lake occupancy could represent periods of high growth for Cisco. And Dr. Tvester's team is looking into measuring each eye layer diameter and pairing these with data on age and growth information from the otoliths to better understand patterns observed. In the coming years, we'll be able to present some more information specific to that portion of this work. But with that, I'll say thank you for listening today, and you're welcome to reach out to me via email anytime with questions. Thank you.